Hello everyone, I'm Michail Chatzianastasis and I'm working as a research scientist at Natera. Today I'm excited to present you Deep Antigen Sets, our new deep learning framework that predicts promising neoantigens for personalized cancer vaccines. Personalized cancer vaccines are designed to teach patients' immune system to spot and attack tumor-specific mutations. But for this to actually work, we need to find effective neoantigens that can truly trigger a strong immune response. The challenge is that many previous machine learning approaches analyze peptides individually and rely on sparse laboratory data such as binding assays or T-cell activation tests, which serve only as imperfect proxies for the true immune response in patients. Results from in vitro experiments do not always translate directly to the complex environment within a patient, where multiple neoantigens may compete for presentations, dominate the immune response, or be overlooked by the immune system entirely. Therefore, our approach, deep antigen sets, instead of scoring each peptide in isolation, treats the entire set of neoantigens from one patient as a single holistic input. In other words, given the patient's whole repertoire of potential neoantigens, we train the model to predict if the patient will respond to IO therapy. Technically, deep antigen sets uses a deep sets neural network architecture. This is a special type of a permutation invariant neural network that can handle input sets of any size, since every patient's tumor has a different number and combinations of mutations. The model first encodes each peptide in a high-dimensional vector using a multilayer perceptron. Then it uses a smart pooling mechanism that weights the contribution of each peptide and combines all of them into a patient context representation. The final prediction head then uses this context representation to estimate the probability that the entire neoantigen sets will elicit an effective immune response. For example, whether the patient is likely to benefit clinically from IO therapy. After training, we can score each peptide individually and obtain a probability score for each neoantigen of the patient. To make this work, we trained deep antigen sets on neoantigen sets from 543 patients who received immunotherapy. Each set can contain thousands of possible candidate neoantigens. We also implemented a targeted sampling strategy during training to focus learning on the most informative peptides, helping to reduce noise from less relevant data. On our internal holdout cohort, deep antigen sets achieved 69% AUC for predicting whether a patient will clinically respond. Then we also tested our method on 3 independent clinical datasets, NCI, Tesla, and HiTide, for neoantigen discovery. The question here is, if you use deep antigen set to rank a patient's neoantigens, how often will you actually catch the truly immunogenic neoantigens on the top of the predictions? The results were very promising. Our model consistently recalls over 60% of the true immunogenic peptides in the top 20 predictions, over 80% in the top 50 predictions, and over 90% in the top 100 predictions. These results outperform previous baselines across all these 3 different cohorts. What makes deep antigen sets stand out is that it captures the contextual interplay between the different neoantigens. Instead of treating each candidate as independent, it can model all the different interactions between them in the patient's immune response. To wrap up, deep antigen sets show that moving from epitope level to patient level learning and modeling the whole set of neoantigens together, we can improve our predictions on new neoantigens. And this brings us closer to the promise of reliable and effective personalized cancer vaccines. Thank you for listening.